Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This is cell 6, which is light microscopes and resolution. Now you might notice, if you've kept the lesson sequence that I gave you in the pre-course lecture on the big 5 questions that simplify biology, which included the list of all the lessons, you might notice this is a bit different. The title on that one was Microscopes and Magnification, but in making the videos I found it was actually a bit bigger than I originally thought, so I split it into light microscopes and resolution, which is this one. Resolution is a really important concept, and then I've also made a cell 7, which is electron microscopes and magnification. So you've got one extra video than initially planned, but it's just so they're in smaller, easier chunks for you to follow. It's still aimed at GCSE level though, and as always, please do subscribe if you haven't done so already. Thank you. Okay, so let's have a look at microscopic measurements. Now obviously cells are too small to be seen with the naked eye. Almost all of them are. There's only a few that are large enough to be seen by the naked eye. So what that means is that we need some different measurements. We can't rely on the normal measurements that we've been used to, millimeters, centimeters, meters and things like that, because the objects we're looking at are just way too small. Those smaller units then come from dividing down the units that we have. So you're all familiar with a millimeter. You know that on a normal 15 centimeter ruler that you might have in school, you've got 15 centimeters. Each centimeter is divided into 10 millimeters. Well, almost everything in the cellular world is much smaller than a millimeter. So we have to look at something called a micrometer. Now, a micrometer is 1/1000th of a millimeter. So there's 1,000 micrometers per millimeter. And the symbol for micrometers is this. It's that little mu symbol from the Greek, and then the M for meter. Okay, when you're coming to draw that mu symbol, easiest way to do it is to just do a U and then put a little line down from the U. You can see that looks like it. That's all you need to do when you're writing it. Now, even though we've broken a millimeter down into 1,000 increments, we still need to break it down much further for a lot of the things in the cell. So we then look at 1 micrometer, and that equals 1,000 nanometers. And the symbol for nanometer is just NM, so that one's very straightforward. Now, if you do the maths there and look at how a nanometer relates to a millimeter, it's 1 millimeter is 1,000,000 nanometers, because there's 1,000 micrometers in a millimeter and 1,000 nanometers in a micrometer, so it's 1,000 thousands, which is 1,000,000. So 1 millimeter is 1,000,000 nanometers. And to give you a little bit of perspective on that, let's think about some usual measurements, some general things. That an average animal cell is around 20 microns long, okay, or 20,000 nanometers long. So 20 microns, think, given that there's 1,000 micrometers in a millimeter, micron and micrometer are the same thing, by the way, sorry to abbreviate. If it's 20 micrometers, that means it's 20/1000ths of a millimeter, or to put that another way, it's 2% of a millimeter. It's absolutely tiny. That's your average animal cell. But even then, think about something really small. The cell membrane, the skin around the outside, is only 7 nanometers thick. That's 7 1/1,000,000ths of a millimeter. These are the kind of sizes we're talking about. It's absolutely ridiculously small. But that's why we need these measurements. You need to know those figures above me there. You need to know exactly how those different things relate to one another. But let's give you a few more examples. If I pull up a picture of a cell here, and let's recap what was in cells. 1, these 2 key structures are vital for having a cell. The membrane makes it a compartment separate to everything that's outside, and the cytoplasm fills it up. So as long as you've got membrane and cytoplasm, you've got that container. You then need to have chromosomes of DNA, because those are the instructions for how to build protein. And you need the ribosomes, because those are what's going to build your proteins. And remember, proteins do everything. Okay, so as long as you've got those 4 things, you can have a functional cell. Let's throw in some of the other things that are in a normal eukaryotic animal cell. We've got the nucleus, so a nuclear membrane there to protect the DNA. We've got mitochondria. It's the mitochondria that will do the aerobic respiration to provide energy for the cell. And let's throw in a small temporary vacuole there. So those are all the main things that we talked about that are in an animal cell. Let's look at their sizes now. We've already said that the cell membrane is 7 nanometers thick. So actually, on my diagram, it's very misleading, because to make it obvious, I've put the cell membrane as quite a thick line on there. So actually, let's just thin that out, and it may just be that very, very thin line. And even what I put on there to that scale is still too thick, really. Okay, but it gives you a better idea. Let's look at something else now. Let's look at the nucleus. The average nucleus, and if you remember to the mitosis presentation, the nucleus does grow a bit as the DNA turns from single to double chromosomes. But on average, a nucleus is 10 micrometers diameter, 10,000ths of a millimeter there. So that's really, really small. But look at the DNA that I've got in there as well. You can see those chromosomes. Well, DNA is actually too small to be seen. So actually, put more realistically, the nucleus looks a bit more like that. Okay, I thinned the line of the membrane around the nucleus, and I've just made it a bit darker. You can see there's a darker patch in the middle because actually the DNA is so small and so spread out that it just looks like a bit of a misty thing inside the nucleus. So it looks a bit more like on that picture there. Mitochondria, 2 microns long. Now, if you look at the diagram, you look at the nucleus, and we've said that's 10. Mitochondria are 2, and we need to change our diagram again. Mitochondria would only, at the scale of the nucleus, be about that big. They're really, really small. But hopefully you can see why I drew them so much bigger. It's so you can see the detail. It'd be very difficult to see any detail on mitochondria of that size. Okay, ribosomes. They're only about 30 nanometers wide. They're absolutely tiny. So actually, I'll shrink them down on my diagram now. You can see I've just put them as tiny little dots. But even then, they're too big. Ribosomes really would not be visible to the naked eye on this diagram. Okay, but we'll put them as those tiny dots just so they're there, they're represented. And the small temporary vacuole is about 3 micrometers. So that's about correct at that scale. But you can see, looking at that cell, it's really not very easy to see what those things are. So that's why generally most cell diagrams aren't to perfect scale, just because they're diagrams they're trying to help you to understand and learn. So they enlarge things that they need you to see. But that gives you an idea of some of the relative sizes of things that are inside a cell. You may be asked to quote some of those in the exam, or they might refer to them in the exam. So it's useful to get a rough idea of how the sizes of those different things relate to one another. So given how small all these cellular objects are, humans have invented ways to look at them. Now, the light microscope was discovered a very, very long time ago, and the earliest microscopes were very, very primitive. We're talking several hundred years ago. Very, very primitive. You'd put a dot on there and you'd try and hold it up and you'd look through tiny, tiny little lens. They're much more advanced now, and you should be familiar with using a light microscope. Hopefully you've used it when you were a bit younger through the school. You may have used it as part of your GCSE course in a bit more detail. But this should look pretty familiar to you. The standard bench lab light microscope. Let's just add a few labels there because you've got to know what the different bits are. But firstly, it's called a light microscope because it relies on the human eye, which can only see visible light. Okay, that's light from sort of 400 to 700 nanometers wavelength, if you want to link this up to physics and the electromagnetic spectrum. But it's a very, very narrow band. And because the light microscope that relies on the human eye, it's called a light microscope. It's not called a light microscope just because it's got a light on it. It's because it uses light. Okay, so let's look at the different parts of it. You've got the base at the bottom and the arm on the side. Those are just standard parts. The base to hold it steady on the bench, on the tabletop. The arm just to connect the top pieces. Really important bit though, you've got the stage. Stage, just like in a theatre, people go on stage to do their performance. It's on the stage of the microscope that the performance is happening, because that's where you put your slides. Okay, whatever you want to look at, whatever your sample is, you put it on a microscope slide, which I put a picture of in the top there, and the slide goes onto the stage to be viewed. You hold that slide on the stage by the clips. Now the clips can vary depending on the type of microscope. Some of them, like these, hold it from the sides and help to move the slide around. Some of them just clip on top of it to keep it in place, but there's always going to be clips on top of the stage. You've then got the really important bits. You've got the objective lenses. Those are the lenses that come down that have got the glass lenses in them that will magnify what is on the stage, what's on the slide. So those objective lenses are really, really important. But at the top, you've also got an eyepiece lens. It's those two together that give you your magnification. And magnification is just how many times bigger something looks. So between the objective lens and the eyepiece lens, it's how much bigger can they make the object look. So if you're looking for the overall magnification of a microscope, you need to multiply together the objective lens and the eyepiece lens. So for example, if your objective lens was 40 times magnification, it made things look 40 times bigger, but then your eyepiece lens was 10 times, then it's 10 times bigger than the 40 times. So it's 40 multiplied by 10. So the overall magnification of that microscope with a 40 times objective lens and a 10 times eyepiece lens would be 400 times. So you're making the sample on your slide look 400 times bigger than it is when you're viewing it with the naked eye. Okay. Carrying on to a couple of other points, you've got how you make the image look clear. You've got the coarse focus dial, that's the bigger one, and the fine focus dial. All those two things do is move the stage up and down, closer or further away from the objective lens to bring it into focus. Okay, coarse focus moves the stage quite fast, so you can get it up to, oh, that's roughly correct now. Then use the fine focus to make the tiny last little adjustments just to get it exactly perfect for your eyes. Okay, those are really important key bits of the microscope. Last couple of bits. It has got a light. I know it's called a light microscope, but it's a light microscope because it uses light, not because it has a light. But nonetheless, you've got a light on the bottom which is going to shine light up through the sample. Okay, you've then got underneath the stage, you've got this thing called an iris diaphragm. That can just kind of open or close like that to control how much light goes through your sample and up to your eye. Because you can adjust the light because that will help you get a really clear image. I suggest take a quick screenshot of that just so you've got all the key parts of a microscope, because you will need to know those. Let's have a quick look at what a light microscope can do then. Let's look at a few photographs taken through a light microscope. So this one over here, it's a fairly standard picture of plant cells. You've probably seen these in class. You may have done this experiment, just taken a very thin layer of onion cells. You can literally peel it off, put them on your microscope slide and look through them. And you can see the different parts there. If you're struggling a bit to identify what you've got, I've labelled up a cell there. There's many cells on that picture, but that bracket label just marks one out for you. Each one of the cells on there, you should be able to identify its nucleus. And like I said in the previous slide, you can't see the individual strands of DNA in there when the cell's in its normal stage of its life, stage 1, okay? Because they're too spread out. You can just see it as a sort of darker blob in there. You've got the cytoplasm that fills up every cell, and of course, because these are plant cells, you should be able to identify the cellulose cell wall there. That's that darker bit in between all the cells, and you can see it creates a sort of physical barrier between the cells, because you've got the cell wall of one cell and the cell wall of the cell next to it, making quite a wide bit of cellulose. Okay, let's pull up another picture. Oh, sorry, and the cell membrane. If you look really carefully, just inside the cellulose cell wall, you can see the dark line, very thin dark lines. That's the cell membrane, but you can identify that on a light microscope. And remember, we said that was 7 nanometers thick. So light microscopes can see really quite microscopic things if they're pretty obvious. Okay, let's look at animal cells now. This one's been stained, so you can see the nuclei much more clearly on this one, and it's at a slightly higher magnification than the previous one. But again, you can see a cell there. You can pick out lots of cells on there. The dark purple blobs, the nucleus inside each cell. So you can see the nucleus in these cells. These are skin cells. Takes up a lot of the room there. You can see the cell membrane around the edge, and you can see cytoplasm inside all the cells. So again, a light microscope allows you to look at cells, but you can't look at much detail inside the cell. Of course, you don't have to just look at cells through a microscope. It's just a random example here of a flea. Okay, fleas, tiny little things. You can see them with the naked eye. If you've got cats or dogs, you may have seen fleas before, but they are ridiculously small. But if you've got one, a dead one, and you put it on a microscope slide, you can put it under a light microscope and you can have a really good look at it, and you can see the hairs on its legs. You do get a lot of detail with a light microscope, but not really enough if you want to make a detailed study of cells. Now at this stage, we really need to look at why light microscopes can't give you a really good image. And it's because of this thing called resolution. Resolution is a vital thing when it comes to viewing anything, because magnification alone isn't enough. Now, we already said that magnification is how much bigger you can make something, how much bigger the image is compared to the actual object. But resolution is how clear and detailed the image is. Now, if you've got an unclear image, magnifying it isn't going to help. It's just going to magnify the blurriness. Let me show you some examples to make sure you understand that. So here's a photograph I took last year when I was up in Scotland. You can see a butterfly in the middle there on a thistle. It's a nice photo. You can see in the background, you can just about see on the one side that you're looking out to sea beyond, that there's a rock. You can see some white surf banging against it. It's a nice photo. But if I grab that butterfly and I magnify it, that's the image we get. That's not a very good image, because that photo is not at a very high resolution. It's not very clear, not very sharp. So when you magnify it, it kind of magnifies the blurriness. So you can see there's lots of pixels there that I took. So this was taken on my camera, which is 24 megapixel. That's a very high resolution photo. To get the first one, I just used a digital package to reduce the resolution. So this is the proper high resolution image, but it's the same picture. Okay. If I now magnify the butterfly, totally clear. You can see all the details on his wings. You can see all the little hair-like fibers on the wings. You can see his tongue sticking out to get the nectar out of that thistle. You can see the antenna up on top. Compare the two oval pictures that you've got. This one, very high resolution. That one over there, not high resolution at all. So you need high resolution. You need to have captured lots and lots of detail if you're going to magnify it and still make it visible. If you don't capture that detail in the first place, magnifying it is pointless. So there's no point magnifying an image that was taken with poor resolution. And a microscope with really high resolution can capture much, much smaller objects, meaning they're still visible to the human eye when they're magnified to be big enough to be seen by the human eye. Okay. Now, a microscope's ability to do that is called its resolving power. Ideally, you want microscopes with very high resolving power to make really sharp, detailed images so that when you magnify them, you're magnifying that detail. If it was low resolution and it never captured the detail, magnifying it isn't going to reveal that detail that was never there. Okay, now as I said at the start of this video, I have had to split microscopy down into two videos. So what you need to do now is go straight on to Cell 7, which completes this bit and looks at electron microscopes, which have a much, much higher resolution. So we look at what those can do. And it looks at the maths of the magnifications. So head to that one next. There should be a link coming up just over there. Thanks a lot. If you haven't subscribed already, by the way, please do so. There should be a little circle on my face above my head. Just click on it if you haven't already subscribed. Like the video if it's been useful, otherwise head straight to Cell 7 on electron microscopes. Thank you.